Hello, my name is Joshua Mugabi. I welcome you to Cell Theory. Today, our lesson objectives are functions of life, cell theory, microscopes, magnification, surface area to volume ratio, differentiation, gene packaging, multicellular organization, cell differentiation, and stem cells. So we are going to start by looking at the functions of life. All living things carry out 7 basic life functions. We are saying all living things, they carry out 7 basic functions, 7 basic life functions. And they're the ones that we want to look at today. The first one is metabolism. And this one undertakes essential chemical reactions. All the essential chemical reactions, they fall under metabolism. The second, we have reproduction. And this one produces offsprings. They can be sexual or asexual. Sexual is use of gametes, and asexual, there is no use of gametes. Those are basic life functions. We have sensitivity. This is responsive to internal and external stimuli. Changes that are occurring inside, they are an internal stimuli. And those ones that are occurring outside, they are the external stimuli. So one of the 7 basic life functions is sensitivity. That is to respond to the changes in the internal and external environment. We have homeostasis. Now, homeostasis, this one is the maintenance of a stable internal environment. Stable internal environment. You keep the internal environment stable. It can be in terms of pH, can be in terms of concentration, temperature. All those ones, if they are maintained at a stable state, that is a life function, and it is homeostasis. We have excretion. This is the removal of toxic waste products from the body. Removal of toxic waste products from the body. We have nutrition. The exchange of materials with the environment. Our body is exchanging materials with the environment, taking in nutrients and then removing out undigested materials. That is nutrition. Then the 7th basic life function is growth and movement. This is the change in shape, change in size, and change in position. Some organisms can move from one place to another, which we refer to as locomotion. But others, They change position by growth. For example, the plants. They cannot move from one place to another, but they can change position by growth. Then we have other organisms, animals, and so on. Those ones can move from one place to another. So those are the 7 basic life functions of all living things. Now, we can use mnemonics to remember them. Mr. Sheng. This one represents M, metabolism, R, respiration, reproduction, S, sensitivity, H, homeostasis, E, excretion, N, nutrition, and G, growth. So we can use those mnemonics for us to remember the 7 basic life functions of living things. Life function examples. We want to look at 2 organisms, the Palamecium and Chlorella. And we are going to use our mnemonics to see how these life functions occur in the 2 organisms. We start with metabolism. The Palamecium uses heterotrophic type of metabolism, and Chlorella uses autotrophic, feeding by making its own food. Palamecium, in terms of reproduction, it is asexual reproduction. That is under mitotic division. The same applies to the Chlorella. They still reproduce by asexual reproduction using mitosis. When it comes to sensitivity, the Palamecium uses chemotaxis. For example, moving towards food. That's how it senses, using chemotaxis. Then the Chlorella is going to use phototaxis, moving towards light. In terms of homeostasis, both of them, they keep an equilibrium. When it comes to excretion, the Palamecium is going to excrete via the anal pore, whereas the Chlorella will use diffusion. And when it comes to nutrition, the Palamecium has got food virtues, whereas the Chlorella carries out photosynthesis. Then growth and movement. Palamecium moves via cilia, whereas the Chlorella is non-motile. It does not move. We want now to look at the cell theory. And this one describes the structure function or structure organization of all living organisms. It is describing the structure organization of all living organisms. According to the theory or the cell theory, living things are composed of cells. Living things are composed of cells or their cellular products. 2. The cell is the smallest unit of life. It is the smallest unit of life. 3. cells only rise from pre-existing cells, meaning that cells come from existing cells. Now, there are some cell theory exceptions which are contrary to the cell theory. Certain types of cells and tissue do not conform to the standard notion. They do not conform to the standard notion of cells. Those are some cells and tissues. For example, we have striated muscles. Here you find that individual cells fuse to form long multinucleated fibers. Yes, we are saying a cell is the smallest unit. But here individual cells are fusing up and they are forming a long multinucleated fiber. A giant algae. Now, certain species of giant algae are very large. They are very large. Yet we know that a cell is the smallest unit of a living organism. When you look at a septate fungal hyphae, here you will find that the hyphae may be connected by a continuous cytoplasm, meaning that the cell now is becoming very large, which is contrary to the cell theory, that the cell is the smallest unit of a living organism. Let's now look at the cell scales. In science, objects are typically measured in metric system. We use metric system in science to measure objects. We have units, prefix, and symbols. If the unit is 1, we can use meter. If the unit is 10 to power negative 3, we can use millimeters. The unit is 10 to power negative 6, that is micrometer. 10 to power negative 9, that is nanometer. 10 to power 12, that is picometer. So, we have meters, we have millimeters, micrometers, nanometers, and picometers. So when I'm moving from 1 meter to a micrometer, I'm going to divide by 1000 and I will get 10 to power negative 3, which is a millimeter. When I divide a millimeter by 1000, I'm going to get a micrometer, so which is now 10 to power negative 6. If you get a micrometer and divide it by 1000, you will get a nanometer, which is 10 to power negative 9. And from a nanometer, we divide by 1000, we get a picometer, which is in the size of 10 to power negative 12. So those are our scales. Now, cells and their components are usually measured in micrometers and nanometers. When we look at the membrane, it is in 7.5 nanometer. That is its size. If you look at the size of a bacteria, it is 1 micrometer. You look at the size of a paramecium, it is 100 micrometer. Now, when you look at these three components or cells, components of cells, you will see that the bacteria is measured in micrometer. Paramecium cell is also in micrometer, meaning that the paramecium cell is bigger than the bacteria. But again, when you look at the membrane is measured in nanometers. So that means that the bacteria is smaller than the paramecium cell. And again, of the three, you will see that the cell membrane is smaller than the bacteria and smaller than the paramecium. Now, when we look at the molecules, anything that we call molecules, these ones are measured in nanometers. If you look at the virus is also measured in nanometer, 100 nanometers, meaning that it is bigger than the molecule. If you look at the organelles, like for example, the chloroplast and mitochondria, they are measured in micrometers, 10 micrometer. So when we compare the three, the molecule, the virus, and the organelles are bigger because they are measured in micrometer compared to the virus. And also the virus is smaller like the molecules, but the viruses are bigger than the molecules. So we are saying that the cells and their components, we usually measure them in micrometers and nanometers. Now we want to look at the microscope. We want to look at the microscopes. We know that objects that are too small for the naked eye may be visualized with microscope. Anything that is small, that cannot be seen by the naked eye, the only way we can see it or visualize it is by using microscopes. And we have the light microscope. The light microscope, this one views living specimens. It views living specimens, and these specimens are seen in their natural color. The way they appear. For example, if you look at this organism of ours here, this insect, it is in its natural color. So if we want to see it in its natural color, we are going to use a light microscope. But a light microscope has a low resolution. It has a low resolution. Now, when we are talking about resolution is whereby you can distinguish 2 points between two organelles which are very close. If you can separate the two organelles from each other, that is what we call a resolution. So you find that a microscope, the light microscope, has got a lower resolution. And even its magnification is also very low. So if you look at the bacteria, when you're looking at the bacteria using a light microscope, you are going to see them with a lower resolution. You cannot separate a bacteria from another bacteria, but you will see them in the light microscope. Because of the low resolution and low magnification, we are not going to see those points separated. And when you look at the plant cell, of course we shall see the plant cell, but we shall not be able to see the organelles very clearly inside the plant cell. But we can use still the light microscope to see the cells. The same applies to the animal cell. We can see the animal cells, but we cannot see clearly the organelles inside the animal cell. That's when we are using a light microscope. And even the muscle tissues can be seen, but still we cannot see clearly the organelles or the organs that are inside, because the light microscope has got a low resolution and it has got low magnification. Now, another type of microscope is the electron microscope. Now, the electron microscope, for it, if we are using it, it views dead specimen. And these dead specimen are viewed in monochrome. That means that if you look at your structure here, the organelle of your insect here, you can see that it is dead. The electron microscope is used to view specimens that are dead, which means these organs have to be dead if we are to use an electron microscope. And the second thing is that we are just going to see them in a monochrome, not in their natural color, like the way we see using a light microscope. But an electron microscope has got a higher resolution. It has a higher resolution. That is a light microscope. And it has also got a higher magnification. So we can easily separate between two organs that are close together. That is a resolution, higher resolution. And it can magnify the object clearly for us to see, meaning that it has got a higher magnification. Now, if we look at the bacteria seen under an electron microscope, you can see that now the bacteria can be seen separated, meaning that the electron microscope has got a higher resolution and it has also got a higher magnification, that the bacteria can be seen one by one. Yet they are very small compared to the light microscope. And if you look at the cell here, it can show you the virus in green, and then we have the bacteria which is in red, and then the animal cell which is the other part which is blue. So here you can see that you have a high resolution. There is a very high resolution that we can clearly see the bacteria, we can clearly see the virus, and we can clearly see the animal cell. Very high resolution and very high magnification. And if you look at the plant cell, you can see that now in your plant cell here, there are certain things that you can see. For example, here we can see this one here. We can see a chloroplasts, Those are chloroplasts. We can see the vacuole in the center. We can see other organelles very clearly seen. So that means that the electron microscope has got a higher resolution. The only disadvantage is that we see the images in monochrome, not in their natural color. Let's now talk about magnification. We have linear magnification over drawing or image. That one we can call it MIA, where we shall say that magnification is equal to the image size divided by the actual size. That is when we are carrying out linear magnification. We can use MIA. Magnification, you get the image size and divide it by the actual size. Then we have the actual size of the object. Actual size of the object. The actual size of the object. If we want to calculate the actual size of the object, we are going to say the actual size is equal to the image size divided by the magnification. So we are still using MIA here. And we are just interchanging and making the actual size as the subject. So when we get the actual size as the subject, we shall say image size is divided by the magnification to get the actual size. That is MIA and A. Let's look at a worked example. Calculate the magnification of the image. Calculate the magnification of the image. Now, when you look at this image, it is giving us a distance here. From this point to that point, it is 50 micrometers. And if you look at the total length of the whole insect or the whole image is 13 centimeters. So what are we going to use? So we shall say magnification is equal to image divided by the actual size. Our magnification is equal to the image divided by the actual size. So our image, if you look at our image, is this whole block. From here to here, that is our image. Here to here, which is 13 centimeters. And 13 centimeters, if we are to change them into micrometer, then we shall get 13 divided by we are saying we want to get the image size. So we are going to get the 13 centimeters and convert them into micrometers. But to change the 13 centimeters to micrometer, what we are going to do, we are going to get the 13. We are going to multiply it by 10 to power 4. And that one will give us 130,000 micrometer. That one is going to be our image size. Then after getting our image size, we need the actual size. Now the actual size, we are going to get 50 micrometers multiplied by 7. And then divide, this one will give us 350 micrometer. Now someone may ask, where are you getting the 7 from? Where are we getting the 7? Now, if you look at our organism here, and you measure from its actual size. Here they are saying from this point to here, it is 50 micrometer. But if you are to measure from here and go across this organism up to where it stops, we shall see that there is 1, 2, 3, 4, 5, 6, and 7 from the total actual size is 7. And if they are 7, but one of this one is 50 micrometer. So those 7, we shall multiply them with the 50 micrometer. And it is the one which is going to give us the actual size, which is 350 micrometer. Therefore, our magnification is going to be our image here divided by the actual size. And our image, we know now that it is 130,000 micrometers. So we shall come and put it here, 130 micrometer divided by our actual size, which is 350. And this one is going to give us a magnification of 370 micrometer. That is going to be our actual size, or the magnification is going to be 370. Let's talk about surface area to volume ratio. Cells need to produce energy to survive, and this requires material exchange. If the cell is to survive, there is need of energy. And this energy is going to come by exchange of materials between the cell and its environment. Then our cell will be able to get energy and survive. So, the rate of metabolism is a function of the cell volume. Rate of metabolism is a function of the cell volume, meaning that the extent at which metabolism is going to take place is going to rely on the cell volume. Therefore, the rate of material and heat exchange is a function of the surface area. Here what we are saying is that the rate at which materials are going to be exchanged, materials and heat are going to be exchanged in and out of the cell is going to depend on the surface area of the cell. But earlier we have said that the rate of metabolism, the rate at which metabolism is taking place, is going to depend on the volume of the cell. Remember at the beginning we have said the cell needs energy in order for it to survive. And if it is to survive and to get this energy, there has to be exchange of materials. So how are we going to exchange the materials? We have to exchange materials in the cell and out of the cell. But that one is going to depend on the surface area. Therefore, the metabolism is the one that is going to give us this energy. And it is also going to depend on the size or the volume of the cell. Now, as a cell grows in size, volume increases faster than the surface area. So we can see that when the cell is changing in size, its volume goes on increasing. And as it is increasing, the volume is increasing faster than the surface area. So this means that as the volume is increasing, there is decrease in the surface area to volume ratio. So we shall have a decrease in the surface area to volume ratio as the cell is growing in size. Its volume is going to increase faster than its surface area. So volume is increasing, surface area to volume ratio is decreasing. Now, if the metabolic rate now exceeds the capacity to exchange material, the cell is going to die. Now, metabolic rates are the reactions that are taking place in the cell. And when these metabolic rates are increasing, or they come to a point that they exceed the capacity of exchanging materials, the cell is going to die. First of all, we have inputs, like nutrients are going inside. And when they go inside, metabolism is going to take place. And when metabolism is taking place, we are going to produce energy. And if this energy is produced, it has to come out of the cell. But if the rate at which the metabolism inside is greater than the capacity of exchanging materials, putting in nutrients, and removing out of products, then the cell is going to die. So there has to be an equilibrium with the metabolism and the rate at which materials are entering into the cells and the rate at which products are coming out of the cell. So those ones should have an equilibrium with the rate of metabolism if the cell is to survive. surface area still to volume ratio. Growing cells typically divide and remain smaller in order to survive. When cells are growing, they go on dividing, and this division helps them to remain small so that the cells can survive. So the surface area to volume ratio is important in the limitation of the cell size. One, why is the cell growing and continuously dividing? Is to increase the surface area to volume ratio. And why are we increasing the surface area to volume ratio? So that there can be an equilibrium between metabolism taking place inside the cell and the exchange of materials, nutrients going inside and products being produced should come out of the cell. So if we have a lot of metabolism taking place inside the cell faster than the rate at which nutrients are coming in, then the cell is not going to survive. It is going to die. If we have a lot of products being produced than the products that are going out of the cell, then the cell is also going to die. So what should happen so that we maintain an equilibrium between metabolism and exchange of material is to increase the surface area to volume ratio. Therefore, that one is important in the limitation of the cell size. So the size of the cell should remain small, and that one helps it to have a higher surface area to volume ratio, which is going to help the cell to exchange materials at a faster rate. That is in equilibrium with the metabolism that is taking place inside the cell. Now, surface area increases, but the volume stays constant. How? When one cell divides, keeps on dividing, the higher the cells divide, we increase the surface area to volume ratio than having all the cell making one block. If it is one block, the surface area to volume ratio is low. But if we divide all of them into smaller units, then we increase the surface area to volume ratio. So when we look at the total volume we say the height times the width times the length. If you have from here up to here is your height, and from here up to here is your width, then from here to here is your length. When we multiply those ones, they will give us the total volume. Now, if you look at this big box of ours here, its total volume is 125. But when you divide it into smaller boxes and we get our volume here, the total volume also remains 125. Now, if we look at the total surface area we shall say it will be the length times the width or times the number of sides of the box. Now, here what are we getting? You will see that the total surface area of this one is 150, our big box cell. And for the smaller ones, the total surface area is 750. So what does it imply? That now our surface area to volume ratio, our big box is having 1.2. Whereas, if it is divided into smaller cells, the surface area to volume ratio increases to 6. So we can see that the surface area increases when we subdivide the bigger box into smaller box. The surface area increases, but the volume remains constant. So, if you look at these cell tissues, that we have here, they have a higher surface area to volume ratio. How? If you look at these cells here, all those ones, they are villi, increasing in size, which means instead of having only these cells down, they put there these prolonged ones. The prolonged ones are going up. All these prolonged ones, they are increasing the surface area to volume ratio. Therefore, those ones increases the area where exchange of materials takes place. These are microvilli, increasing the surface area. So, we want now to look at multicellular organisms. Multicellular organisms, these ones are formed when groups of individual cells function together. Groups, they are groups of individual cells. They function together, meaning that we have very many cells coming together and forming a group and then functioning together. For example, we have the muscle. If you look at the muscle, when the muscles come together, they are going to form a tissue. For example, we have the cardiac. Now, these ones, when they come together, they will now form an organ. And when the organs come together, now we form our system, cardiovascular system. Now, this one becomes now a multicellular organism. Starting from the cell, cells coming together forming tissue, tissues coming together forming organs, and other organs coming together forming a system, and then we form our multicellular organism. Let's now talk about cell differentiation. Multicellular organisms have properties that emerge from the interaction of their cellular components. What we are saying, we have organisms that are made up of very many cells. And these organisms have got properties. But those properties of multicellular organisms, they come from an interaction of their cellular components. We have seen that a multicellular organism is made up of very many cells coming together to form a tissue. Many tissues come together to form an organ, many organs coming together to form a system, and we form our multicellular organism. Now, the property of that multicellular organism comes from the emerging interaction of the individual cellular components. Now, we see that all cells in multicellular organism share an identical set of genetic instruction. All cells in a multicellular organism, they share identical sets of genetic instruction. We have looked at the cardiac structure. Now, all those cells, they are sharing identical sets of genetic instructions. Why? Individual instructions, they are called genes. Those individual instructions, we refer to them as genes. And the genes form a totality which we call a genome. A genome is the genome number or addition, summation of all the genes that we find in an individual. And those are the ones that we call individual instructions. They are the individual instructions. Genes, they are the ones that have got the instructions. And the collection of all the genes in an organism is what we call a genome. Now, the activation of different instruction in specific cells will cause these cells to differentiate and become specialized. I want us to understand this one. Activation of different instruction in specific cells. What is it going to do? It is going to cause these cells to differentiate and become specialized. Now, when we are looking at this one, we are saying all the cells have got the same genetic instruction. They have the same genetic instruction, same genetic information. But here we are looking at a cell going to differentiate. It is going to specialize. By specializing, we are saying it is doing a specific function, possessing a distinctive functionality. How do they get this? Yet they have the same genetic instructions. This one is achieved by activation of different instruction in specific cells, meaning that the genetic instruction, it is going to be activated for that particular group of cell. The activation will be at a specific point on the particular genes are the ones that are going to be activated in those types of cells. And then we shall say that the cell is differentiating. So we have identical cells. These two identical cells, they are going now to possess different gene expressions, but they were identical, and all of them, they have got chromosomes, and these chromosomes have got specific genes. And if these specific particular genes are expressed, gene A here is expressed, and here it is gene B which is expressed. And when that one occurs, here we shall get a different type of cell, because gene A was the one that was expressed. And in here, we shall get another type of cell, because it was gene B which was expressed. But all these cells came from identical cells. That is what we are calling differentiation. They are coming from the same cells with the same instructions. But when it comes to activation, different instructions are activated in specific cells, and those cells are the ones that are going to, particular genes are the ones that are going to be expressed, and then we shall form a specialized type of cell. Let's talk about gene packaging. Now, within a nucleus, active genes are packaged in an expanded and accessible form called the chromatin. So remember, we are saying in our nucleus, it's where we have our genes. But now all these genes are not going to be expressed for that particular type of cells. So when the genes are packing themselves, those ones that are going to be active, they will be expanded and they will be accessible. And that is what we call euchromatin. We have our set of instructions, the genes on the chromosomes. But all those genes are packaged. They are packaged like the way you pack things in the box. But now, they are those boxes that you want to use, particular boxes which have got items you want to use at that particular time, are the ones that you are going to open. So it is the same that is applying to our packaging of genes. When these genes are packaged in the chromosome, those ones that are going to be used are the ones that are going to be expanded, and they are the only ones that are going to be accessible. Those are the ones that we call the active genes. Therefore, the inactive genes are packaged in a condensed and inaccessible form called heterochromatin. Those are inactive genes. They are inactive genes. They are not going to be used by that particular cell. Therefore, they are packaged in a condensed and inaccessible form. So they will not be tampered with. They will not express themselves. And those ones that are packaged in those particular cells, specific cells, they are the ones that we call heterochromatin. Those are inactive genes. Those ones that are going to express themselves in that particular or specific cell are the ones that we call euchromatin, and they are the ones that will be expressed. So now when we look at our structure here, we have a nucleus micrograph. This is a magnification of our nucleus, and inside we can see the white and black. if you look inside, you will see some spaces which are white, and then you will see also those other spaces which are black. You can see those ones that are black spaces. Those black spaces that you are seeing, they are the ones that we are calling heterochromatin. Now, those ones are the ones that contain inactive genes. They are packaged very strongly. They are not going to be accessible. Then these ones that we have, the white ones here, white areas that we are seeing here and there, those ones are exposed, are unpackaged. And that's where we have our genes that are going to express themselves, or what we call active genes. So, differentiated cells have different regions of DNA packaged according to their specific function. So when we have cells that are specific, and we are referring to them as differentiated cells, they have different regions. They are different regions on the DNA that is packaged according to their specific functions. So we are saying that particular type of cell, it is specialized, and inside its DNA, it is packaged in such a way that those ones which are active genes are packaged in a loose way that they are accessible. And those ones that are not required for those particular cells are packaged in a condensed way, which we call heterochromatin. They are inactive. So, differentiation involves the expression of some genes and not others in a cell genome. Cell genome, we are saying it is the general collection of all genes, and all these ones are found in a cell, but all of them are not expressed. So in differentiation, some genes are going to be expressed, others will not be expressed. Let's look at the stem cells. Now, stem cells, these are cells that are unspecialized. These are cells that are unspecialized, are the ones that we call the stem cells. And they have two qualities. One is self-renewal. They can continually divide and replicate. What we call stem cells, they can divide continuously and replicate. Secondly, they have potency. That is, they have the capacity to differentiate into specialized cell. So, a stem cell is a cell which is not specialized, but it can continue to divide and replicate, but it is not specialized. That is what we call self-renewal. And the potency, they have that capacity to differentiate into specialized cell type. That is, they can change into any type of cell. Now, we have embryonic stem cells. Embryonic stem cells. These are totipotent or pluripotent. They can form any cell type. Whereas adult stem cells, they are multipotent and unipotent. These ones have limited capacity for differentiation. We are giving you the different types of stem cells. One is the embryonic stem cells, the potpotent and pluripotent. These ones, stem cells that we find in the embryo, they can form any cell type. Whereas the adult stem cells that we find in adults, that we are calling multipotent and unipotent, these ones have got limited capacity for differentiation. They have limited capacity for differentiation. Self-renewal, meaning that they can continue to replicate and divide. Potence means that they can differentiate and turn into different cells. Those are the stem cells. You have a stem cell that can continue to renew itself, dividing and replicating, forming other stem cells. And then its potence, it's whereby that particular cell can form for us different specialized cells or tissues. That is potence. Renewal, it can continue to divide and replicate itself. Potence, it can specialize and give us different types of cells or tissue. Now, the capacity of the stem cell to divide and differentiate is necessary in embryonic development. We are talking about now embryos. Now, an embryo starts with one cell. And that cell, we are now referring to them as stem cells, which means that these cells should be able to divide and be very many stem cells. And then also, they should be able to differentiate if the embryo is going to develop. And all of us, we know that when a sperm meets an egg, they form what we call a zygote. And now that one, we have one cell, fully one cell. And that cell starts what we are referring to as self-renewal, dividing, becoming very many cells. Now these many cells will later on differentiate what we are calling potence. They are stem cells, the same cells with the same genetic information, instruction, but now they have that capacity to differentiate and give us different types of cells. For example, they can differentiate and give us the red blood cells. They can differentiate and give us different cells. They can differentiate and give us muscle. They can differentiate and turn into nerve cells. That is what we are referring to as potence. And it is very important in embryonic development. Stem cell theory. Stem cells can be used to replace damaged and diseased cells. We can use the stem cells to replace damaged and diseased cells. So when the stem cells are got, they can be harvested from the long bone. And when they are put in the laboratory, they can be made to differentiate into red blood cells. And these ones now can be implanted. So stem cells are extracted from an appropriate source. And one of the appropriate sources that we can get the stem cells, they can be the embryos, umbilical cords, we can get from blood, and certain adult tissues. We can extract the stem cells from there. And after they are extracted, biochemical solutions will trigger cell differentiation. We provide biochemical solution. You extract them from the bone, and then you provide them with biochemical solution. These biochemical solutions are going to trigger these stem cells to differentiate. And when they trigger them to differentiate, then we are going to get new cells, which can be implanted in the host's tissue. These stem cells have been got from the bone. They have been given biochemical reactions which trigger cell differentiation. And they have now turned from stem cells into red blood cells. Now these ones can be implanted in the host's tissue. So stem cell therapy can help us to replace dead cells or diseased cells. Now, the immune system is suppressed to prevent rejection, because now these ones are coming from stem cells which are coming from the bones, and you're going to implant them into the host. There is the immune system is going to try to reject them. So that one can be suppressed to prevent rejection. The immune system of the host can be suppressed so that it does not reject these new cells that are coming. So there has to be cell monitoring to ensure that they don't turn into cancerous. The cells have to be more monitored. These new cells, they have to be monitored so that they do not turn into cancerous cells. Therefore, the capacity of stem cells to divide and differentiate makes them suitable for therapeutic use. Stem cells, because they have the capacity to divide and differentiate, that is making different cells, they are very good to be used in therapeutic use. Examples of stem cell therapy. We have Stargardt disease. This is an inherited form of juvenile macular degeneration, which can lead to blindness. And it can be treated by replacing the dead cell within the retina with a functioning one. And that one is an example of stem cell therapy. Stargardt disease, which is a juvenile macular degeneration, and that one can lead to blindness. But that one can be treated by replacing the dead cells within the retina with a functioning one. We have the Parkinson's disease. This is a degenerative disorder which is caused by the death of dopamine secreting cells. It is a degenerative disorder caused by death of dopamine secreting cells or the central nervous system. And this one can be treated by replacing the dead cells in the brain. You replace the dead cells in the brain with the functioning one. That is Parkinson's disease. That is found in the central nervous system. Ethics of stem cell use. Now, we look at the different sources. We shall look at the growth potential, tumor risks, harvesting, and then disadvantages. The first one we have the source is the embryo. Now we are looking at ethics of stem cell use. Meaning the source here, we are going to get these stem cells from the embryo. Growth potential. The growth potential is high, which we refer to as pluripotent. What of turning into tumor risk? Also that one is the highest. How can it be harvested? It can be generated artificially by somatic cell transfer. Disadvantage. This one means that there is a requirement of destruction of the embryo. Destruction of the embryo. This results in the loss of potential life. That is the disadvantage of getting stem cells from the embryo. Then we have another source, which can be the umbilical cord. Umbilical cord blood. That is going to be the source of the stem cells. Growth potential, it is a little bit low, multipotent. What of the tumor risks? It has lower risks. And how is it harvested? This one is easily obtained and stored or preserved from the umbilical cord. What is the disadvantage? Cells must be stored from birth at a cost. This raises an issue of financial accessibility, meaning that these cells, when they are got, they must be stored from birth. So we have to keep them, which is going to call for financial accessibility. Then we can have another source, which are the tissue from adults. We can get stem cells from tissues of adults. Their growth potential is multipotent, it is low. Tumor risk, it is lower risk. What of harvesting? We can have invasive to extract. Invasive to extract. And the disadvantage may be restriction in scope or availability. So that is ethics of using stem cell. Hello, my friend. Thank you for participating in cell division. We have looked at the functions of life, cell theory, microscopes, magnification, surface area to volume ratio, differentiation, gene packaging, multicellular organisms, cell differentiation, and stem cell. Thank you. Don't forget to subscribe.